All right, so today we're going to start by picking up where we left off. We've been looking at this idea of the central dogma, which involves DNA replicating so that cells can divide. It involves transcription, which is what we focused on Tuesday, which is how um, within cells, a temporary copy of DNA is created. That temporary copy is called messenger RNA. It's made out of a type of nucleic acid that's a little bit different from DNA, but it's not super different, it's just a little bit different. And that messenger RNA can move from where the DNA is to where proteins are actually built. The building of proteins is called translation, or sometimes we call it protein synthesis. Usually protein synthesis has to do with transcription as well, but a lot of times people use all of these terms somewhat interchangeably because the end result of a protein being created using the information stored on DNA ends up being the same. So we're going to look at translation in a little bit more detail today, and there's kind of three main pieces to it. We'll look at these a little bit closer up in just a second. The first is that the messenger RNA gets interpreted by a different kind of RNA called transfer RNA. This is transfer RNA right here. This weird loopy molecule is transfer RNA, and this is actually pretty close to what it looks like. This is sort of stylized, but it's pretty close to what it ends up looking like. Transfer RNA is made of ribonucleic acid, just like messenger RNA, but it's shaped differently and it does something different. tRNA is used to transfer information from the messenger RNA to the ultimate building blocks of proteins, which are called amino acids. The tRNA links to the messenger RNA, and all of that is enclosed by this relatively small glob of stuff called a ribosome. Ribosomes are made out of RNA and proteins together. Ribosomes are not just one kind of biochemical compound. They're made out of RNA plus proteins all together. Ribosomes are kind of like an enzyme. They facilitate this chemical reaction, but because they have RNA in them as well, they're not always referred to as a pure enzyme. Pure enzymes are generally considered to be proteins. These ribosomes are associated with amino acids. Amino acids are the building blocks of proteins. The amino acids get hooked together, and a long chain of amino acids is a protein. So as the transfer RNA and the ribosomes work together to move along the messenger RNA and figure out the information that's there, those amino acids get hooked together to form a longer and longer and longer chain of amino acids, which is a protein. Where the structure of all of this comes in is that the information in DNA and the copy of messenger RNA and the transfer RNA that reads the copy of messenger RNA all uses the same code. And the code is what's translated in the process of translation. The code is a 3-base sequence. And we have 4 different bases. With a 3-base sequence, there are 64 different possibilities of arrangements of letters. We could have 3 A's, we could have 2 A's and a C, we could have 2 A's and a G, we could have 2 A's and on our DNA a T. We could have 1 A and 1 C followed by an A and a C followed by a C, et cetera, et cetera, et cetera. You end up with 64 different possibilities when you look at all the different ways we can arrange 4 base pairs into groups of 3. Those are the codes. That's the information in DNA. And translation uses the structure of DNA, messenger RNA, and transfer RNA to interpret that code. transfer RNA we'll look at in a little bit more detail here, but a lot of the stuff that we're looking at is not of critical importance. We don't care about these loops on the side, for example. They're here. It's just not that important. What's really important about transfer RNA are the ends of it. Okay, so what happens with transfer RNA is it's a strand of ribonucleic acid, just like messenger RNA. But those nucleotides are hooked together in such a way that instead of just being a long strand, it makes kind of a loop. So if we were to look at this transfer RNA, here's the strand, and then it makes a loop here, and it comes back, and it makes a loop here, and then it comes back, and then it starts like kind of hooking onto itself, and it makes another loop here, and it's hooking onto itself, and it makes another loop here, and it hooks onto itself, and the whole time that it's hooking onto itself, it's using those same base pair rules. You'll have like an A at one part of this transfer RNA strand, and that would hook with a U on another part. But instead of being a double strand like with DNA, it's a single strand that's kind of hooked to itself. And the arrangement in a loopy curved shape means that it takes up less space. And on one end of it, there's going to be an amino acid. And on the opposite end of it, there's a code. So because it's got a loopy shape, it's two-dimensional instead of one-dimensional. We've got two ends, each of which can have information and stuff stuck to it. This end down here has 3 RNA bases, 3 RNA nucleotides. They could be any 3. Those 3 nucleotides will be complementary to a set of 3 nucleotides on the messenger RNA, and they attach. So you can see here that on this transfer RNA, there's a C that hooks to a G on the messenger RNA strand. So these are complementary. The U and the A are complementary, the C and the G are complementary for this particular transfer RNA. We have names for the 3 bases. When it's on the messenger RNA, we call it a codon. And when it's on the transfer RNA, we call it an anticodon. This is kind of getting back to what we were talking about on Tuesday, where we're sort of flipping back and forth. If you were to look at the original double strand of DNA here, what you might notice is that you're going to have 2 strands of DNA. One of those we used to make a complementary strand of messenger RNA. That complementary strand of messenger RNA is now read by a complementary transfer RNA strand. So we have like a bunch of sort of mirror images. If we call this strand right here strand 1, then this strand of DNA is strand 2. The messenger RNA here that's made, though, is just like strand 1. So this is just like strand 1. The anticodon on the transfer RNA looks just like strand 2 of the original DNA. We call strand 1 the coding strand. We say that's where the information is, and it matches what's on the messenger RNA. We call strand 2 the template strand. That's how the messenger RNA is built. It's built along that template strand, complementary to the template strand, and that ends up being the same as what shows up on the anticodon of the transfer RNA. On the other end of the transfer RNA, there's going to be an amino acid, and that amino acid is different for different anticodons. We have 64 possible anticodons. So potentially we could have 64 different amino acids. Turns out we don't have that many. Some anticodons are associated with the same amino acids. You can have multiple anticodons and multiple codons that are associated with the same amino acid. But on the other end of the transfer RNA, we have an amino acid. That amino acid is predictable. If you tell me what anticodon you have on a transfer RNA, then I can tell you which amino acid is attached to it. That anticodon will always be associated with the same codon. That's always going to be associated with the same section of the template strand of the DNA. And that's always going to be associated with the same coding strand of the DNA, which means if you tell me what the 3 bases are for DNA right here, if I know that this is a G and then an A and then a G, then I know that my template strand of the DNA is going to be a C and a T and a C. And I know that my messenger RNA is going to have a G and an A and a G there, just like this one does. And I know that my transfer RNA anticodon is going to have a C and a U and a C, which is basically the same as our template strand. But RNA doesn't have thymine, it has uracil. So instead of a T, it has a U. So it's very predictable. It's this flip-flop mirror image. And it doesn't matter if you remember which part of the mirror image is associated with which particular piece. The only thing that matters is that you understand that it's predictable. You show me 3 nucleotides in DNA, we can figure out what nucleotides will be on the messenger RNA, what will be on the transfer RNA, and what amino acid that'll be associated with. This is what we call a codon table. Codon tables are generally written so that they tell us what amino acid will be associated with an mRNA codon. So this is a table of mRNA codons. Okay, and we could figure out what the coding strand, the template strand of DNA would look like, and what transfer RNA would hook onto these codons because it follows the base pairing rules. But those codons are 3 letter sequences, 3 base pair sequences that determine which amino acid will be on the end of a transfer RNA. And you can see here that we have a number of codons that end up giving us the same amino acid. These 3-letter abbreviations here, PHELEU, ILENET, VAL, these are amino acids. Okay, so these are amino acids, the building blocks of proteins. These are amino acids. These are amino acids. These are amino acids. You don't need to know the names of these. The only thing you need to understand from this diagram is that these are abbreviations. LEU, for example, stands for leucine. MET stands for methionine. GLU stands for glutamine. So you don't have to know those names. You just have to know these are the different amino acids and that these are just abbreviations. The capital 3 letters are the codon. Okay, so these are the different codons. And if you looked at a sequence of messenger RNA and you saw, oh, it's got a G and then a U and a U, then you would know that that is going to attach to a transfer RNA that has a complementary anticodon, so that would be CAA, and that transfer RNA would be associated with VAL, or valine, the amino acid. There are some special codons. AUG is a special codon. AUG always does 2 things. One is it's associated with a transfer RNA that's going to be UAC that has the methionine amino acid on it. The other is that is the start of every protein. Every single protein starts with methionine. So we call that the start codon. So AUG, which is the codon for methionine, is the start codon. And then we have a few stop codons too. The stop codons don't also instruct the ribosome, and they're not associated with transfer RNAs that have amino acids attached to them. The stop codons, which are UAA, UAG and UGA, those just cause translation to stop, and that's it. They don't also attach an amino acid. There's only 20 different amino acids, kind of have extra codes. And you can think about this in a couple of different ways, but what's most important to recognize here is, just like all the other stuff we've been talking about for the last several days, there's no conductor. This is not planned. There isn't somebody orchestrating all of this stuff. It's not that there was somebody who said, oh, hey, let's make a code where we can have 64 different possibilities, but only use 20 of them. It's just that this is what happened. And this actually makes sense when we think about why it is that there might be a little bit of redundancy here. But I'm going to let you all puzzle through that a little bit later today. So there's 20 different amino acids that all get hooked together to make proteins. Right. So if we think about how this all works, starting with the double helix of DNA, basically what's going to happen is we're going to have a double strand of DNA that's going to be located in eukaryotic cells in the cell's nucleus. We're going to have a RNA polymerase start making a messenger RNA strand that will be complementary to one strand of the DNA. That's the template strand. The coding strand will look just like the messenger RNA. That messenger RNA is going to have a 3-nucleotide sequence somewhere on it that starts with A, U, and G. That's where the protein is going to start. The process of protein synthesis happens when we get this ribosome. And ribosomes really are like blobs. So this really kind of looks a lot like what a ribosome looks like. You can actually see ribosomes in light microscope. They just look like little blobs. They're very nondescript. The ribosome attaches to messenger RNA, and then inside the ribosome, there's space for 2 different transfer RNA molecules. This right here is a transfer RNA molecule. See how it's all weird and loopy? This one is drawn a little bit differently than the other one was, and that's okay. It's still got the essential features of transfer RNA, which is there's an anticodon at one end and an amino acid at the other end. Okay, so that was for one transfer RNA. For this transfer RNA, here's the anticodon, and here's the amino acid. Within the ribosome, the 2 amino acids associated with the 2 transfer RNAs that are in the ribosome get hooked together. That bond, as some of you probably remember, is called the peptide bond. And then the ribosome moves a little bit further along the messenger RNA. It ejects one of the transfer RNA molecules, which now no longer has the amino acid on it, and it pulls in a new transfer RNA with a different amino acid. Every time the transfer RNAs that get pulled into the ribosome will have an anticodon that is complementary to the codon that's on the messenger RNA. We can even see here, this is going to be a codon-anticodon pair that are complementary to each other. The base pair rules determine which transfer RNA molecules are going to fit and attach to the messenger RNA strand. Okay? So this is a lot. We've done a lot with DNA over the last several days. What we're going to do now is you have a couple more videos that I'm going to ask you to look at. There's an Amoeba Sisters one. There's a Professor Dave one. The reason that I'm having you watch the videos, in addition to the recorded presentation that you're watching right now, is because a little bit of repetition and some animation can be really useful. with this. And then after you've watched the videos, there's 2 assignments. I want you to wrap up the transcription and translation simulation activities. And there's another assignment called Translation POGIL. And some of you may notice that that's actually designed for AP biology. It's pretty challenging, but I think actually it's well within your skill set. You all are at a point where you are capable of thinking at that higher level that it would take for you to be able to understand that assignment. So I'm going to ask you to work on both of those assignments, get as much as you can done, get stuff turned in. Enjoy your afternoon, and I will see you